Hello everyone, this is Harshini, Department of Pharmaceutical and Analysis, School of Pharmaceutical Sciences, VISTAS. Today's my topic is Pharmaceutical Analysis. The introduction of pharmaceutical analysis is, it is the branch of practical chemistry involved in the process of identification, determination, quantification and purification of substance, separate the compounds of the solutions or mixture or determination of structure of the chemical compounds. The pharmaceutical analytical scope. The main scope including the quality control, analytical method and validations. The quality um, the quality of pharmaceutical products in pharma industries, QC and analytical preparators. Types and techniques of pharmaceutical analysis. There are two types of chemical analysis, qualitative and quantitative. Qualitative means involve the various procedures or designs for the identification of compounds. Identification of compounds in sample. The test result to confirm the presence and the absence of the compounds samples to the analyte. The techniques of pharmaceutical analysis is titration analytical, spectroscopic analysis, chromatographic analytic, electrochemical techniques, separation techniques, thermal techniques. The chemical techniques used in qualitative analysis. The qualitative analysis involved in identification of ions or functional groups in the compounds, the various chemicals and the physical techniques. For example, solubility, melting point, boiling point, density, specificity, density and specificity, gravity, refractive index, viscosity, optical rotation, limit test. The chemical techniques used in qualitative analysis is inorganic and organic qualitative analysis. The inorganic qualitative analysis is systematic method of analyze to confirm the presence of cation and anion present in an inorganic substance or mixture of a substance. Organic qualitative analysis of the compound is generally heated in air solid using melted they burn with either the smoky steam in some back reduced of the carbon. Some compounds are present in carbon, hydrogen, oxygen, nitrogen and metal ions. Next one is solubility. The solubility is defined in qualitative terms of concentration of soluble in concentrate of solution at this certain temperature. The solubility is not used for identification, but it improves the information for the compound which is used to analyze. Next one is melting point. The melting point is the temperature which is a soluble is converted to the liquid at normal atmosphere pressure. A more specific definitions of melting point, the temperature at the solid and liquid phase or the equilibrium at the specific pressure. For example, glucose, sucrose. And fourth one is boiling point, the temperature at which the boiling vapor pressure of the liquid equal the pressure surrounding the liquid and the liquid changes into a vapor. Example, water boil 100 degrees Celsius. Next, density and specificity gravity. Nothing but density means that the mass per unit volume of the substance, but on the earth you can assure the mass in the same as weight. However, the specific gravity is expressed at different temperature. It will longer be equal to the density. For example, chloroforms. Next one is refractive index. Nothing but it is the characteristic property for the identification of organic solvent, liquids and solution. Any impurities in this sample will affect the refractive index and hence it is used to qualitative measurement. Seventh one is viscosity. The viscosity is defined as a measure of resistance of the fluid to deformation by the stress. In other words, to Viscosity described as a liquid resistance to flow. When the intermolecular force of the attractions are sum within the liquid, there is a large viscosity. The eighth one is optical rotation, optical activity and specific rotations. Nothing but this is an important property helping to determine the purity of mass. Most of the sugar, pharmaceutical chemicals and liquids and molecules too can rotate the plane or polarized liquid light. The property rotation the plane to the rotated light is called optical activity. The optical rotation is dependent in the concentration, path length, temperature, wavelength of the liquid used as a solvent. Ninth one is limit test. Limit test is nothing but qualitative or semi-qualitative test designed to identify and control the small quantities of impurities which is likely to be present in the sample. The target and impurity present in a substance in a small quantity. The impurities like heavy metals, iron, sulfate, chloride, etc. The importance of standardization, the impurities of the substance. Next one is quantitative. Quantitative analysis. Nothing but the category of analysis including quantitative and determine the compounds in the sample to generate the quantitations as between done to based in the same physical properties of the compound. The quantitative analytical technique are further classified as follows. The follows as a technique is chemical method, phytochemical methods or instrumentation methods, electrical methods, chromatographical method, hybrid technique method and biological and microbiological methods. First method is chemical method. Chemical method is nothing but volumetric or titrating method. First one. Second one is gravimetric method, gastroanalysis and electrogravimetric. First, summarize to the chemical methods are based in chemical reaction. The volumetric method measure the volumes of the standard solution. Gravimetric method measure the precipitate and the gastro method measure the volume of gas. Nothing but the volumetric methods or titrating method to involve the substance to determine the approaches reagents of the standard solution and the volume of the solution to complete the reaction is determined. The volume method to require the simple and less the apparatus and that the high accuracy. And the gravimetric methods, nothing but the substance to be determined is converted in the insoluble precipitate in the purity form, which is that corrected and weighted. Gastroanalysis to involve the measures of volumes of the gas involved or observed in a chemical reaction. The sum of the gas which are analyzed the gastrometric or the helium etc. Phytochemical methods or instrumental methods. The instrumental methods involved in measurement of some physical properties of the compounds or the substance. These methods are employed for determine the minor or track concentrations of element in the substance. The instrumental methods are selectivity, highly speed, accuracy and simplicity of analysis. Any changes in the properties of the substance to determine the measurement of observed specific rotations of refractive index, margin difference, characters of mass, ratio, etc. The two methods of instrumental methods are spectroscopic methods and emission methods. The common spectroscopic technique is UV, IR, etc. And the common emissional technique is frame photometry, mass spectrometry, etc. And third one is electrical methods. The electrical methods nothing but involve the measurement of electric current, voltage, resistance in the reaction to concentrations of some specific in the solutions. For examples, potentiometry, conductometry, polarography, amplitudometry, voltmetry. Next, the chromatographic methods. Chromatographic method is nothing but the chromatography technique and the electrophoric methods or separation methods for the mixture of the compounds, but also applied for the identification of compounds of mixture. The various chromatographic techniques are GC, HPLC, TLC, HPTLC, etc. And the fifth one is hybrid techniques method. Nothing but the above the technique produce the hybrid technique. The several examples to proper used today and the new hybrid techniques are under developed LCMS and GCMS. Common hybrid technique is LCMS, GCMS, LCMS by MS, GCMS by MS, ICP-MS. The advantages is highly sensitive, highly selectivity and more reliability result, save time and samples. The system is biological and microbiological methods. Nothing but biological methods are used to when the drugs to drive we cannot do proper determine and any physical and chemical methods. These methods are based on the responsible of living organisms parts to the substance being the test. Under the microbiological, nothing but observe the potential of antibiotic and microbial agent. The antimicrobial assay the inhibiting of growth of bacteria by the sample is compared to the starter antibiotic. The differentiate of qualitative and quantitative. Qualitative means identify the chemical nature or present in the substance in a samples, and qualitative means determine the amount of concentrations one or more substance in the sample. So the what is the presence in the sample, how much is present in the samples. Nature of the result is describe the given gives identified nature of the substance result is numerical of the measurable values is non-numerical in qualitative analysis, quantitative is numerical. The qualitative analysis in detecting of fluoride, AgNOT tests identify the functional groups, metal ions. The quantitative is NaCl titrations and ion samples concentration of glucose in blood. Methods to use in qualitative is precipitation, flame test, TLC spectroscopy identifications, and qualitative is method to use titration, gravimetry, spectrophotometry, chromatography, etc. And accuracy in qualitative analysis is relative less accuracy, and accuracy is quantitative is highly accuracy and precision, and qualitative analysis is to identify and confirm the presence of compound proposals, and quantitative is to determine the extra amount and concentrations of compound in propose. And role in analysis is first step to find out what is there, qualitative. Okay, next step quantitative analysis is next step to find out how much is there in the role of analyzed. Thank you so much for your attention and patience. Thank you.